Good afternoon, everyone. Welcome to the 2023-2024 GW Cross-disciplinary seminar series focused on cellular metabolism and metabolites. This seminar series aims to promote networking and collaboration across different translational disciplines to support the advancements in the treatment and prevention of human diseases. This seminar series is supported by the Office of Integrative Medicine and Health and the Department of Biochemistry and Molecular Medicine. Today's speaker is Dr. Vertes, a professor of chemistry affiliate at George Washington University. Since joining GW's Department of Chemistry in 1991, Dr. Vertes has had an incredibly productive career. His research interests have a broad reach and encompass the development of new analytical techniques that have been useful to diverse fields such as chemistry, biology, and medicine. Research areas include single cell and subcellular analysis, high throughput, and ultrasensitive methods that are applicable in systems biology, proteomics and metabolomics, and new methods for molecular imaging of biological tissue. His research has been presented in over 200 peer-reviewed publications, and he's a co-inventor on 19 patents, some of which have been licensed and commercialized by industrial partners. He's received many prestigious academic honors, such as the Distinguished Researcher Award at George Washington, the 2000 Hillebrand Prize, and the Oscar and Shoshana Trachtenberg Prize for Scholarship. Dr. Vertes is also a fellow of the American Association for the Advancement of Science and a fellow of the National Academy of Inventors. Dr. Vertes' research efforts to explore biology at the single cell level have been unique from how many of us explore biology using single cell broad omic approaches. While traditional transcriptomics allow for the amplification of small amounts of molecules, amplification is not possible to study metabolites. So today, Dr. Vertes will discuss how his research to examine metabolites at the single cell level have used a more sensitive approach, mass spectrometry, to make some of these discoveries. Welcome, Ecos.
Thank you, Brett, and thank you for the invitation. I'm going to share my screen now. Share. Okay. Can you see my screen?
Yes.
Okay, wonderful. So, as I said, thanks for the invitation, and thanks for the great introduction. So today, today's talk may be a little bit more on the technical side compared to the previous presentations, but that's what we do in my lab. And the challenge of single-cell metabolomics has already been actually mentioned by Brett, but I'll elaborate on it a little bit more. But before we go there, let's look at the rationale. Why are we interested in analyzing the metabolites in individual cells? In order to have an analogy, I have here a medical analogy that distinguishes metabolic states, because that's really one of our main objectives when we analyze single cells for metabolites, is to understand the differences in the metabolic path, the functioning of the metabolic pathways, and how they differ from each other, and potentially discover the presence of hidden subpopulations. So the medical example I have here is the analysis of a large general population for A1C, the glycated hemoglobin that is commonly used as a biomarker for type 2 diabetes. If you look at the general population and look at the distribution in the middle with the dashed line, that shows what you would observe for a general population that comprises people with and without type 2 diabetes. Now, if you look at the subpopulation of people who actually have type 2 diabetes, we know that the A1C hemoglobin percentage is showing higher values, and you can see that with the solid line here, showing a slightly shifted distribution. So what this means is that obviously the metabolic pathways work different for people with type 2 diabetes. We see that the only way to actually separate the subpopulation with this particular metabolic state is to analyze individuals, not general population, and then pool the blood and get some average value, because that will not distinguish or that will not flag the people who are having this condition. So likewise, when we look at cells, we look at cellular functions. Even if you have an isogenic cell population, depending on various factors, we will see differences in the metabolite levels. And let's assume now, just for a second, that we actually have a technique that can analyze this population of cells individually, one by one, and we can measure metabolite levels in each and every one of these cells. So we can actually work on this data in two different ways, depending on whether we are co-registering with the metabolite levels, we are co-registering the position of the individual cells. The simpler version of the technology is when we just analyze the single cells and we perform statistical analysis and get histograms like the ones you are seeing here in the lower right corner, where on the horizontal axis you see the concentration of a particular metabolite, and the vertical axis shows you the cell counts, the number of times you are encountering a particular concentration for the metabolite. As you can see, this histogram shows a bimodal distribution, which can be an indication of two different subpopulations. And it also shows what you get when you actually measure just the average concentration. That would be the bulk value for the concentration. And you can see that the bulk measurement is obviously missing the two subpopulations, but it also misses the actual values, the highest values for the concentrations for the highest populations for a particular concentration. So this points out why single cell analysis can reveal the hidden phenotypes that may be in the system. Now, the other pathway here shown on the top is when we co-register the location of the cells, and that enables us to actually reassemble images of the distribution of a particular metabolite in the cell population. So this type of omics is called spatial omics, when you learn not only the metabolite levels, but also how it is distributed in the cells within the tissue. So, as Brett has mentioned, we don't have the ability to amplify metabolites. The same with proteins. We can't amplify proteins either, unlike, for example, transcripts. So that means from the analytical perspective, we have a major challenge, which means that we only have the metabolites available in that individual cell. And that cell, depending on which kingdom we are talking about, here I have to compare the microbes and plant cells. They can be very different in their volume. The volume is characteristic of the amount of metabolites available. So, for example, a yeast cell is about 30 femtoliters in volume, whereas an Italiana cell is 20 picoliters in volume. Now, the corresponding metabolic models show diversity of genes, reactions, and metabolites, as well as compartments, depending on how advanced the model is. The reason I'm showing these numbers here is that we have an idea how many metabolites do we need to target, do we need to detect and identify in a cell, in a single cell, to get an idea of the metabolic pathways. So, for example, in this Italiana cell, in this particular model, works with over 1,700 metabolites. Another interesting aspect of single-cell analysis is that if you think about cells in isolation, it's very different from cells being embedded in a tissue and functioning. So in one of the studies we have done, we looked at Limnia stagnalis, the great pond snail, and its nervous system, central nervous system, and we targeted an individual cell, a so-called FGP neuron, with a known function in this this is a relatively small nervous system, only 20,000 neurons are present. We were able to analyze not only the neuron, this targeted neuron, but separately analyze the cytoplasm and the nucleus. So, but we are not going in that direction now. Because I still need to explain all the challenges associated with single-cell metabolomics. We have covered no amplification. Then there is the volume challenge that our sample is in the femtoliter to nanoliter range. So it's a very, very small amount of sample. Even just the handling of such a small volume is a challenge. Then, of course, there is the limit of detection challenge. Many of the metabolites are present in relatively low concentration, and then you have a small volume, so the overall amount of the metabolite available in the cell is exceedingly small. We have seen the multi-species challenge. We're talking about hundreds and thousands of metabolites in a single cell. Another interesting challenge, also different from transcripts, is that metabolites can have very fast turnover rates, meaning that, for example, ATP in a bacterial cell turns over within a few seconds. So if we don't stop this turnover, or we don't come up with a technique that is very, very fast, we are not going to be able to measure the concentrations and the abundances of metabolites as they exist in the living cell. I've mentioned the tissue embedded cell challenge, so that there is that. So overall, this all points in the direction of one technology, and that's mass spectrometry. Mass spectrometry is very sensitive and is capable of high specificity. So we need to look in that direction. And because of these large variations in volume, we need to develop new sampling techniques and new ionization techniques. So the first technique I want to mention here is a capillary microsampling in combination with electrospray ionization and ion mobility separation mass spectrometry. So I'll break this down now because it's a lot of different technologies that may not be familiar. So the capillary microsampling is a fairly simple method. You take a sharp capillary, you insert it into the cell of interest and extract a tiny bit of the cytoplasm and then use that for analysis. The way that works is that we apply a high voltage to that sample and produce ions that are sprayed into the inlet orifice of the mass spectrometer. Inside, we have a device called ion mobility separation that first sorts the ions based on their physical size, and then so geometrical size. And then these separated ion packets move into the mass spectrometer that further sorts them according to their mass to charge ratio. So unlike with conventional mass spectra, where you have a horizontal axis with the m over z and the vertical axis with intensity, for such an instrument where you also have ion mobility separation, you get a mobilogram that has the m over z on the horizontal axis, the drift time on the vertical axis, the drift time in the ion mobility system, and you get a three-dimensional spectrum where the intensity is color-coded. So we are going to use this technology to look at single hepatocytes, and in particular, the relationship between cellular energy and cell death. So as you know, depending on how much energy a cell has, which is basically the level of ATP, the amount of ATP in that cell, there are three possible pathways for cell death. Apoptosis is the programmed version of cell death. Necrosis is just the obliteration of the cell. And autophagy is when the cell goes into a pathway where the components, the cellular components, are being reused by the organism, by adjacent cells. And the interplay between the damage or stress on the cell and the energy level will determine which pathway is going to be utilized in cell death. So, as I said, we are going to work with human hepatocytes, and we use a capillary. This is now showing the hepatocyte here. The capillary is inserted and extracting the cell cytoplasm, and we pull back the capillary. The cell is detached, but that doesn't matter because we already have the sample in the tip of that capillary. We are talking about tenth of a picoliter of cytoplasm, so very, very small amount. And then, so I said we use electrospray. The way that works is that we apply high voltage to the sample, and that high voltage creates tiny droplets with a lot of charge on every droplet. And then as those droplets fission, we get to the point when individual cellular metabolites are ionized. And here I show now the first example of such a mobilogram, where the horizontal axis is the M/Z value, the vertical axis is the drift time in the mobility device. And as you can see, it separates a lot of different components. Very beneficially, the electrospray background is separated out from the metabolites and from the lipids that are in different parts of this mobilogram. And then if we are pooling the various drift time values, for example, for the lipids, we showed the corresponding conventional mass spectrum here with a very, very nice signal-to-noise ratio. And likewise, if you are pooling the different drift times for this group of points, we get the metabolite spectrum that shows the ADP, ATP, UDP, and hexose, acetylamine, and so on. So altogether, we identified 22 metabolites and 54 lipids from this very, very small sample from a single cell. Now, I told you that we are going to explore cell death in a single cell. So in order to do that, we measured the ADP, ATP, ADP, and AMP levels from the single cell and formed this ratio that's called adenylate energy charge, which is basically a relative metric of the energy state of the cell. This energy metric varies between 0 and 1. When you're close to 1, the cell has sufficient amount of energy and it's a healthy state of the cell. But when you are degrading the energy production in the cell, you get much lower values. So we compared hepatocytes that were raised in regular media, and then separately we spiked the media with rotenone, which is known to disrupt the electron transport chain in complex 1. So what that means, if you add the rotenone, the ATP production in the cell will be degraded. And that means that this adenylate energy charge will be significantly reduced. So that's what we show here in the histogram on the lower left corner, that shows that the DMSO control group of cells shows AAC values averaging around 0.82, whereas in the presence of rotenone, we see this gray histogram that has an average of 0.16 adenylate energy charge. So clearly, we were able to detect mitochondrial dysfunction using single-cell analysis in hepatocytes. Okay, so that's great, but as you probably can guess, a big challenge or a big the efficiency of this technique is its very low throughput. So you need to have a really patient graduate student or somebody in the lab who is willing to sit there and analyze cells one at a time. That's no way to go, especially when you compare this technology to the already mature single-cell transcriptomics, when they are talking about thousands of cells analyzed in a very short time. So we set out to increase the throughput of our technology, and for that we turned to a laser ablation technique that was developed in my lab. It's a mid-infrared laser ablation that we named laser ablation electrospray ionization, or LASI, that we developed for single cell analysis. We developed a version of it that uses an optical fiber that couples the mid-infrared radiation into individual cells and enable us to blow up individual cells and use the electrospray to produce ions from that ablation plume, and then use the mass spectrometry to get the composition, the metabolite composition from that single cell. We also, excuse me, we also set out to automate this process to increase the throughput, and also to do this in a way that enable us to perform cell-by-cell imaging. So that's what's going to be applied going forward. So here is the laser ablation. This is the fiber tip here on the left side. You can see the dark shadow or contour of the fiber tip that is pointing to individual cells, blows up the cell, and produces mass spectra shown here on the top right, typical mass spectrum from a single cell. And if you follow the signal over time for a particular metabolite, you can see the metabolite levels in the various cells as the tip is probing them. So we were able to automate this to the point where on this onion skin sample, this was just used to develop the technology. Onion skin has a very good properties for this. And we were able to analyze over 1,000 epidermal cells in 9 hours. When you have data from over 1,000 cells, you can start to see much better histograms. So you can see for example, the glutamate level here on the top left, the allele level top right, are showing unimodal distributions. These are log normal distributions. But when we were looking at disaccharides, we found that there was a very clear bimodal distribution showing subpopulation of cells with low disaccharide levels and a larger subpopulation of cells with higher disaccharide levels. We did this, of course, for a whole array of metabolites, and in order to characterize the spread of metabolite levels over the population, we introduced the metabolic noise. The definition is shown here. It's basically the standard squared standard deviation divided by the squared mean value is the metabolic noise. And as you can see here in this figure, the metabolic noise shows a very slight dependence on mean value. So that means that the various metabolites have actually quite similar metabolic noise values. As I said, one objective is to discover hidden phenotypes. So we did select here a couple of metabolites that show bimodal distributions in this onion skin. Fumarate, disaccharide, proline, and oxalate are shown here as examples with these two metabolic states, a low and a high level for these metabolites representing these phenotypes. But then we decided, okay, so onion skin is interesting, but let's look at something that's actually of real value. And we turned to a collaboration. We turned to biological nitrogen fixation. And the reason to look at single cells in biological nitrogen fixation, because the root nodules where this process takes place are highly heterogeneous systems. That means that they have a mixture of uninfected and infected cells. The infection comes from GFP-labeled rhizobia in our case. So the rhizobia set up shop in these plant cells in soybean root nodules and start to use the nitrogenase enzyme present in the rhizobia to capture nitrogen from air and make it water-soluble nitrogen into water-soluble nitrogen compounds that the plant can use. So this is, of course, has a great significance for the economy and agriculture. So what we see here is our three images on the right side. The bright field image shows both the infected and uninfected plant cells in the root nodules. The GFP fluorescence image shows only the infected cells. You can see them, they are larger than the uninfected cells. And then when you look at on the right, you see the SEM image, the scanning electron microscope image. You can actually see the plant cells, these large ones, excuse me, filled out with bacteroids. The rhizobia transform into the bacteria into bacteroids in the interaction with the plant cells. And they, we start with uninfected cells here that are somewhat smaller, and then partially infected cells show the plant cell partially filled with bacteroids, but where at the end state, these plant cells are chock full of bacteroids. So that's something we keep in mind for the upcoming discussions. Okay, so we did first manually targeted infected plant cells, uninfected cell clusters, and epidermal cells from these root nodules, and collected mass spectra from each cell type. And then the mass spectra were treated with hierarchical clustering to show the spectral features on the vertical axis here and the cell types on the horizontal axis. And as you can see, hierarchical clustering nicely separates the epidermal cells and their spectral features from the uninfected cells. And not so cleanly, but we can also separate the uninfected and infected cells based on their spectral features. So clearly, for example, these spectral features are present in uninfected cells and not present or slightly present, only slightly present in infected cells. Here too, the problem was that this was still done manually, and we were talking about 100 cells here because of the low throughput. So we set out to improve, further improve the throughput by automation, and also improve the molecular coverage by introducing ion separation by the ion mobility device that we have already seen in the case of the hepatocytes. So the automation is basically shown here. You can see how this is the tissue section from the root nodule. This is the sharpened optical fiber, and as you can see, we are raising the sample and bringing an individual cell in the vicinity of the sharpened fiber tip, and then do the ablation and take the mass spectrum through the ion mobility separation and the mass spectrometer. And now we can, because this is automated, we can get a much higher throughput. Now we are talking about over 800 cells per hour throughput. As I said, the other advantage we were looking for was to take advantage of the ion mobility separation to improve molecular coverage. Here we compare the spectrum from without ion mobility separation and with ion mobility separation. And you can see that the number of spectral features detected has increased almost twofold. So going from 131 spectral features without ion mobility separation, 259 spectral features with ion mobility separation. So, and for example, when you go to the lipid region of the spectrum, you can actually see and identify lipids in the ion mobility separation enhanced spectrum that were not present in the spectrum without the separation. Another advantage of ion mobility separation is improved signal-to-noise ratio. So, for example, here we are comparing the two spectrum with and without ion mobility separation. And this particular part from the overall mobilogram, we can zoom in and look at a very narrow mass-to-charge ratio range. And we can see that there are differences in drift times, but also you can see that the signal-to-noise ratio is greatly improved as a consequence of ion mobility separation. So, for example, while only daidzein, which is a dihydroxide isoflavone, can be identified without ion mobility separation. With the separation, we also detect palmitoleate, a lipid, in the vicinity of this isoflavone. So that is one advantage. Another advantage is that because of the ion mobility separation, we can distinguish structural isomers. So the example we see here is looking at, again, a zoomed version of the mobilogram. Shows here two red dots here with exactly the same M over Z, but different drift times, which convert into collision cross-section. And you can see that here in the drift time chromatogram, you can see the two peaks. So that means we are dealing with a mixture of two structural isomers. And if you look at the potential structural isomers, there are these three possibilities here. The tetrahydroxyflavone glucoside, stilbene, and glucosinol tetrahydroxyflavone. So these three are all possible based on just the mass to charge ratio. But when you combine the collision cross-section measurement with tandem mass spectrometry of this 449.101 parent ion or precursor ion, from the fragmentation we can decide that of these separated structural isomers, only these two are consistent with the tandem mass spectrum. So it really is very helpful when you are dealing with a host of structural isomers to narrow down the possibilities and then use tandem mass spectrometry to nail down those structural isomers. So, but going back to the cellular heterogeneity, we have again created the histograms, this time not from the onion skin cell, but from the large number of cells we analyzed in the soybean root nodule. And here too, we found that certain metabolites in the top row, you can see they are present only with a unimodal distribution, but certain other metabolites, like in this case disaccharide and hexose phosphate and PIP, which is a lipid, were present with bimodal distributions, and indicating the presence of hidden phenotypes, metabolic subpopulation, metabolic states with different metabolic pathways active. So moving forward, there is also another major challenge in metabolomics, not just in single-cell metabolomics, but metabolomics in general, and that is that the mass spectra contain a lot more peaks than we can identify. And the presence of, we are talking about more than 70% of the peaks in a mass spectrum in metabolomics cannot be identified at present. I call this the dark metabolome, analogy of dark energy and dark matter, because it's there, we know it's there, we see there is something there, but we don't know what those things are. So obviously this is a major challenge in metabolomics, and it also is present in single-cell metabolomics. To tackle this, we teamed up with a group at PNNL, Pacific Northwest National Lab. They have a really high-end mass spectrometer with a mass resolution of over 10 million, and mass accuracy is better than 150 parts per billion. So this enables us to identify and resolve isotopic fine structure. In the lower left corner, you see here an isotopic fine structure. These are peaks that are coming from mass shifts, mass differences from isotopes for regular elements. And from the pattern of these isotope shifts, we call this the isotopic fine structure, we can actually decipher the elemental composition of any compound, any unknown compound. So that's what we took advantage of, and we took this to the single cell level. So for the first time, we were able to shoot at a single cell in this root nodule single infected cell, extract isotopic fine structures, and identify unknown compounds, identify known compounds that were also not trivial to identify, and also distinguish compounds that were unresolved in lower performance systems. So here we can see, for example, the isotopic fine structure of N-acetylglutamic acid with the calculated spectrum in showing the red dashed line, very good agreement. In this middle isotopic fine structure shows the distinction between two compounds that were not resolved because they are only 0.6 millidaltons apart in their mass to charge values. And on the right side, we are showing the deciphering of elemental formula for a compound. So with this technology, we were able to identify from a single cell 47 known compounds and discover the elemental composition of 11 unknown compounds. Going back to the abundance distributions for different metabolites, we were again finding that some metabolites showed unimodal distributions. Some were best modeled by normal distributions. Some others were better modeled with log normal distributions. But more interestingly, we also found bimodal distributions, for example, for NAD+, hexose phosphate, and acetylcarnitine. We were interested in what can be behind the presence of these bimodal distributions, in particular in the root nodule, infected root nodule cells. So we came up with a hypothesis and for these hidden phenotypes. In particular, a hypothesis for the NAD+ bimodal distribution of NAD+ here. And the idea was that when we were looking at the scanning electron microscope images of these infected cells, we found that there were cells, plant cells partially filled with bacteria that were obviously still multiplying because we know that eventually all plant cells are completely filled up with bacteria. So we thought about the cell cycle for these bacteria that obviously is going through the complete cell cycle and mitosis for the partially filled cells, whereas the fully infected cells that are volume limited now and the division stops in these cases. So we have the bacteroids in the resting state, the G0 state. And if you compare the NAD+ to NADH ratio for multiplying bacteria and G0 resting state bacteria, you see that low and high ratios are found respectively. So our hypothesis is that what we see is, and what we distinguish in these two subpopulations are the dividing bacteria versus the resting bacteria in the plant cells. Finally, we also used this technology to start developing spatial metabolomics, based on single cell analysis. In particular, we were interested in the analysis of rare cells in a root nodule. So what you see here on the top left is the fluorescence image of the infected cells in a root nodule. Then we took this image and performed image segmentation to identify the infected cells as objects. And then we performed image analysis to pick out a particular subset of these infected cells. We selected these based on cell size, cell volume. So, you see the image, the second from the right shows the selected cells that were relatively large in volume, and we targeted these for analysis. So basically, we took this entire set of infected root nodule cells and selected the large ones. You can, of course, select things based on fluorescence labeling for any number of different properties of these cells. We selected ones that were large. And then we targeted them, we individually analyzed these 25 large cells and measured various metabolite levels like malate, glutamate, disaccharide, and jasmonic acid, and mapped those metabolite levels back onto the cells and their locations. This enabled us to basically show the first targeted analysis of rare cells in a tissue for metabolites. So basically, in conclusion, we developed technologies for high throughput analysis of single tissue embedded cells, and every single attribute here is important because we had to develop separate technologies for high throughput. Of course, we had to develop the technology for single cell, and then we had to also develop a method to do this in tissue embedded cells. There are other techniques in single cell analysis that are based on, for example, microfluidics that are based on dissociating the cells from the tissue. But our objective was to avoid this dissociation step, cell dissociation from the tissue, because we believe that it may lead to change in metabolite levels. We also introduced new technology for improved molecular coverage in the single cell arena. And that was using the ion mobility separation. With that, we demonstrated the improved capabilities. We also, based on these techniques, based on these new technologies, we were able to identify hidden phenotypes or metabolic states at the single cell level. Using yet another technology, the identification of isotopic fine structure at the single cell level enabled us to discover elemental formulas of unknown metabolite or xenobiotics at the single cell level. And finally, using optically guided mass spec imaging of single cells, we were able to show the first example of spatial metabolomics and also target rare cells for analysis. And with that, I want to thank my group, in particular, Dr. Sylvia Stopka and late Samara, who are by now, they have their own career. They were graduate students and postdocs in my lab. Sylvia is at Harvard and late is at Princeton. My current graduate student, Marjan Dolat Moradi, contributed significantly to this. Our collaborators at Pacific Northwest National Lab, Chris Underton and Liliana Pasatolik, were instrumental in the isotopic fine structure measurements on the high-performance mass spectrometer over there, in combination with our front end that we installed there. And at the University of Missouri, we had collaborators, world-class experts in plant biology, Professor Gary Stacy and his student Beverly Aktuka. Funding for this work was provided by the Department of Energy and the National Science Foundation. And with that, I thank you for your attention, and I'll be happy to answer questions.
Thank you very much, Ecos, for that. That was really fascinating and exciting to see so many technological advances all together in one presentation. If anyone in the audience has a question, feel free to either raise your hand or unmute yourself. I know we only have a couple minutes left. While people try to maybe formulate a question or whatever's on their mind, I think it's really neat, the ion mobility separation, increasing the signal to noise ratio and the increased high throughput advances, especially trying to think about how you can, instead of just seeing bimodal states, see a lot more of dynamic changes and predict some of the heterogeneity, or at least uncover some of the heterogeneity. I'm curious, in your opinion, what you think is the next major technological advance that will even help to make that easier?
Obviously, people are really pushing hard now in the single-cell analysis world. And there are the 3 omics that are being advanced. Transcriptomics already has the technology, and it's more or less at this point, it's I would almost call it a routine approach, because it has incredible throughput and it identifies tens of thousands of different transcripts from single cells. Now, proteomics, single-cell proteomics is at a similar level to single-cell metabolomics because of the lack of amplification and the need to use mass spectrometry. They are still pushing for higher throughput, just like we do, and higher molecular coverage. And the same with metabolomics. I think the next big step will be when we can combine the 3 omics at the single cell level, preferably from the same cell. And that will be really exciting, because for the first time, we are looking at a single cell as a laboratory where we can put things back together, the proteins in there and their levels, the metabolites, the corresponding levels, the pathways, the transcripts, and pull the whole thing to the systems biology at the single cell level. And that, I think that's the next big step forward.
That sounds absolutely fascinating. So there was a question that came through as to whether or not this was applicable to mammalian cells, but you did show some hepatocyte work earlier. Quickly, maybe since everyone else is dropping off, to follow up on your answer of integrating all of these omics so much, as there are more of these advances on the technical end of studying metabolomics and mass spec, how much do computational limitations or the computational pipelines form a hindrance for integrating this, or do you think it's more the technology end of just getting high enough throughput and getting deep enough coverage into getting more metabolites and more analytes?
No, you're absolutely right. The computational part is bioinformatics is also a huge part of this, just like it is for systems biology at the bulk level. Now, I would go out on a limb and say that because we're working with a single cell and we are not mixing in pathways from other cells, we may have an easier time to sort things out. The bioinformatics of a single cell may be cleaner than a mixture of all those cells. Of course, when you have a lot of cells, your molecular coverage will be higher. So that's the advantage there. But when you go to the single cell, the pathways that are unusually active or inhibited are showing up more clean in your spectra. And so the corresponding bioinformatics may be somewhat easier.
Great. Okay, so thank you very much, Akos. I really appreciate your contribution to our cross-disciplinary seminar series on cellular metabolism. Best of luck with everything in the future. I'm sure GW will be receiving an announcement as soon as there's a decision made that will be telling us all you have reached emeritus status, and best of luck with all of your other endeavors in retirement.
Thank you very much.
Okay, take care.